Hi, everyone, and welcome to the 2022 PI Conference. We're getting ready to get started here. So everyone knows Spanish interpretation is available for this session. You can access the Spanish language line by clicking the world icon on your Zoom toolbar. I'm Tammy Black, and I'm the Vice President of Communications and Marketing here at the Immunodeficiency Foundation. I'm going to be your moderator. And today I'm thrilled to be joined by Dr. Mel Berger, and he's going to be presenting on the topic of the future of plasma. And I'm going to take just a few minutes to talk briefly about what IDF is doing to ensure that there's adequate access to plasma-derived therapies for PI patients. There will be time for questions at the end of the presentation, so feel free to submit any questions you have to the presenter in the Zoom chat. Mel Berger earned his undergraduate medical and PhD in biochemistry degrees from Case Western Reserve University in Cleveland. After internship and residency in pediatrics at Boston Children's Hospital, he did an allergy clinical immunology fellowship at NIH and is board certified in pediatrics and allergy immunology. He served as assistant chief of allergy immunology at Walter Reed Army Medical Center and on the faculty of the Uniformed Services University of Health Services. That's a mouthful. In 1984, he joined the faculty at Case Western Reserve, where he rose to professor of pediatrics, pathology, and general medical sciences oncology at Case, and chief of the Division of Allergy Immunology at Rainbow Babies and Children's Hospital. From 2008 to 2020, he was the senior director at CSL Behring with roles in research and development and medical affairs, including major efforts in development and subcutaneous IgG and the use of serine protease inhibitors in transplantation. He is now an independent consultant and continues to serve as an adjunct professor at Case Western Reserve. Welcome, Dr. Berger, and we're thrilled to have you. I'm going to hand it over to you to talk to us today.
Okay. Can you see me?
We can see you, yes.
Okay. Well, thank you, Tammy, and thank you all in the audience. It's great to be back at the IDF, even if only virtually. I do have to declare that I'm still doing some consulting for CSL Behring. I've done some for Takeda in the past year and some small biotechs. And I do have stock in CSL Behring, but I don't think any of that is pertinent to my presentation today, which I'll get right into. Okay. Well, okay, you'll have a little other stuff there. So I'd like to discuss the therapeutic uses and values of normal IgG. As far as we're concerned, and most of the industry is concerned, IgG is the most important plasma-derived medicine. And so it's hard to talk about IgG without talking about plasma. And I'm going to try to have a look at both as we go. So we know that the most important mechanisms of action of IgG are its roles against infection. And these are mostly in the line of promoting opsonophagocytosis, which is the engulfing and killing of infectious organisms by white blood cells and macrophages, and neutralizations of viruses, adhesins, and toxins. And this is most important if we have pre-existing antibody from immunizations or a prior infection. Then when we're exposed to a virus or a bacteria that's making a toxin, the antibodies will neutralize it and it won't be able to set up shop. IgG on a target can activate complement, which may destroy the target or may help opsonophagocytosis. Now, it turns out that IgG also has immunomodulatory effects, and I think the clinically most important effect is its ability to saturate a receptor called FcRn, or the neonatal Fc receptor. And this is something on the cells that line our blood vessels and also in the placenta, which grabs onto the Fc, or the handle part of IgG, and returns it to the circulation rather than having it be degraded as most other proteins would be. So IgG has a long half-life, much longer than almost any other plasma protein. And if somebody has autoantibodies, which are causing a problem like an autoimmune disease, and we saturate this FcRn with normal IgG, then the autoantibodies won't be able to be recycled and will be catabolized. And that is probably the single most important mechanism of the use of high-dose IgG for autoimmune and inflammatory disease. Other mechanisms include blocking or preventing the activation of Fc receptors that might be involved in producing cytokines and causing direct cell-mediated cytotoxicity, neutralization of autoantibodies by binding to their antigen binding sites. Excess IgG in the solution can scavenge complement off the surface. This would not be great if we want complement to help us with an infection, but it is quite useful if we want to keep complement from destroying our own cells when we have autoantibodies. And we can also neutralize toxins and superantigens, which is probably the effect we see in Kawasaki disease, which fortunately is not a major problem anymore because we have this therapy, but was quite a dramatic disease before we got IgG therapy. So in the US, not all indications that some products approved carry for all of the products. But amongst the products, we certainly have primary immune deficiency, ITP, which is caused by antibodies against platelets, CIDP, or chronic inflammatory demyelinating polyneuropathy, multifocal motor neuropathy, Kawasaki disease, and dermatomyositis. And I would point out that in most accounts, in most audits, as much IgG, if not more, is used for CIDP than for all immune deficiency combined. Off-label uses common in the US include secondary immune deficiencies, Guillain-Barré syndrome, myasthenia gravis, and other autoimmune diseases, and organ transplantation. Now, in the EU, they have class labeling, which means that all products in the class can be used for all of the indications. The indications in Europe include PIDD, secondary immune deficiency, measles post-exposure prophylaxis, ITP, Kawasaki disease, and the neuropathies. Now, the global usage of IgG is quite impressive. Some of these bars represent 2-year or 3-year periods, but the point is that the overall growth rate of demand or usage of IgG is increasing at about 8% per year, and that continues now even through the time of COVID. So it's a big growth industry, and it means there's a big continuing increase in the demand for the plasma from which IgG is made. I would point out that in around 2020, the use of around 250 tons of IgG globally required something to the tune of 70 million liters of plasma at an average yield of 3.5 grams per liter. And we'll come back to that in a few minutes. Now, a very important point, especially for us in this country or in the US and North America, is that most of the plasma, 2/3 of all of the plasma used in the world, comes from North America, especially the US. And you see relatively little plasma is collected in the rest of the world. And this is mainly because the US allows compensation of donors, but most of the rest of the world, with the exception of a few countries in Europe, does not. So the US is responsible or is collecting much more plasma than we're actually using for plasma-derived medicines like IgG, albumin, hemophilia factors, and so on. Whereas if we look at Europe and most of the rest of the world, even including Canada, they're using more finished products, or the use of finished products for which they account is greater than the amount of plasma they're contributing. And again, this is mostly because these countries use only unpaid, uncompensated donors and recovered plasma, which is the plasma from whole blood that's not used. So we have a continued greatly increasing demand for plasma in the US. We now have something on the order of 400 to 500 plasma collection centers in the US. And it's really incumbent on us as people who require medicines derived from plasma for ourselves, our family members, our patients, to encourage our colleagues, our peers, other people to donate because our lives depend on this plasma, which is mostly collected in the US. So besides increasing donations and collections, how can we sort of use the IgG more efficiently? So in some conditions, especially in chronic conditions, the clinical response can be used to optimize the therapeutic regimen. And this is especially the case in PID, but it's also definitely the case in ITP, MMN, and CIDP. And I'll show you some examples of this in a minute. But the basic point is that if someone has a neuropathy, which is causing them to be weak, then they get IgG and they become stronger. They can tell by how strong they are or how long their recovery lasts, how much IgG they need. Similarly, if somebody has primary immune deficiency, you can tell by the amount of fever or the amount of infections whether you're having enough IgG and you need more, or if you need more. On the other hand, there are some acute conditions in which high dose IVIG is required, but the effect is delayed from the time of administration. So in Guillain-Barré syndrome, the IgG level achieved by the dose that's given acutely determines your ability to walk 6 months later. In Kawasaki syndrome, the dosing is given over a couple of days, but the total dose determines the incidence of coronary artery aneurysms a month after the acute phase. So here you see a collection of patients from Helen Chappell's group in England, and they had patients on a protocol where every 6 months the patients were reevaluated and their dose of IgG was adjusted until they went for the whole 6-month interval with no infections. And then they recorded the trough levels of IgG that were needed to keep them completely free from infection. And you see that whereas some patients could be completely protected from infection at trough levels of IgG of 5 to 7 grams per liter, that in the US would be 500 to 700 milligrams per deciliter. Other patients required more than 10 grams per liter or more than 1,000 IgG level in milligrams per deciliter. Now, this probably relates to the length of time the patient has had the disease, when they were diagnosed, how much chronic infection they have, and to what extent their infections represent flare-ups versus new infections. And of course, it also involves exposure. If somebody's exposed a lot, they may need higher IgG levels to protect against infection. But the point is that it's not one size fits all. And if we adjust people's doses, we can use our IgG most efficiently. Now, this is a study in Guillain-Barré syndrome, an acute paralyzing neuropathy, and they were testing a new formulation of IgG. Everybody got the standard dose of 2 grams per kilogram within days of the diagnosis, and then they measured the levels at different time intervals. So these are the serum IgG levels at 2 weeks after dosing, and they divided the patients into quartiles. So this whisker represents the lowest quartile, or group 1. Here, just below average is group 2, above average group 3, and the highest group is group 4. And if you look now at these patients 6 months later, what you can see is that fewer patients in groups 1 and 2 could walk without assistance than patients in group 3 and 4. So it's, I think, difficult to say that we should trim the dose or cut back the dose of IgG given to patients with Guillain-Barré, because if we cut back the dose, it means that 6 months from now, we're going to have patients dependent on wheelchairs, which if they got the full dose might not be. Similarly, if we look at Kawasaki disease, this is an acute febrile vasculitis, which can result in long-term damage to the coronary arteries. And when I was a student and a resident in pediatrics, we had little babies having crushing substernal pain, crying out at night, and actually having myocardial infarctions. And it was kind of incidentally or serendipitously discovered that IVIG very effectively and quickly reversed this disease. And what was found is that the total IgG dose given over the first couple of days was the determinant of the number of permanent pathological changes to the coronary arteries. So if the babies got 2 grams per kilogram, they had a very low percentage of coronary artery problems, about 5%. But if that dose is cut in half, you see that the prevalence of coronary artery aneurysms, which is a big problem, is also doubles or even goes up higher. So again, cutting back the dose results in increased pathology, and we don't necessarily know that at the time we give the IgG. So one thing we can do is, for those diseases in which we can use the clinical response as an indication of the dose, we can adjust the dose. Now, for some specific infections, we could use alternatives to normal human IgG. For example, plasma-derived hyperimmune globulins, which generally have about 5 times the titer of standard IgG. Monoclonal antibodies and small molecule antivirals, in some cases, may be much more effective than polyclonal IgG, and we certainly saw this in the case of COVID. We also now have the ability to get human IgG from cattle, and we can immunize the cattle and get specific antibodies. And this has been given to some human patients. For autoimmune and inflammatory diseases, we have some alternatives to normal human IgG. These include other kinds of molecules, monoclonal antibodies and bioengineered molecules that can saturate the FcRn and cause catabolism of the autoantibodies that are causing the problem. And we're just beginning to see these receive clinical approval. Now, efgartigimod has now been licensed, and we expect to see some for myasthenia gravis, and we expect more molecules in this class to be able to substitute for IgG in some of those diseases. We can also interfere with the production of autoantibodies by the B cells using drugs like rituximab or many of the drugs that are being developed for B-cell lymphomas. And we now have all sorts of new anti-inflammatory and immunomodulatory drugs, including orally active JAK inhibitors, which may make it possible to spare the use of IgG. But there are some conditions for which polyclonal IgG is indispensable. And as we well know, to us, the most important one is primary immune deficiency. But also for some secondary immune deficiencies, polyclonal normal human IgG is indispensable because we need the range of specificities of anything that might be coughed in our face in a given day. Similarly, for Kawasaki syndrome, we don't have an effective substitute. And for multifocal motor neuropathy, we don't have an effective substitute. Now, very quickly, just to show what happened when we went from standard IVIG to a hyperimmune IgG for the prevention of respiratory syncytial virus infection in at-risk babies, the Respigam required a high dose, 750 milligrams per kilogram, but this was 5 times the potency of regular IgG, which required a huge dose. But then when the monoclonal antibody came along, you only required 15 milligrams per kilogram, which was also much easier for the baby to receive. So here is a dramatic example of how development of a monoclonal antibody led to a real reduction in the use of IVIG. So where do we stand now in 2022, looking at this continued growth rate of use of IgG of about 8%? And it looks by all indicators and market projections as if this will continue, or at least this is continuing to increase at the present time. So in order to satisfy the projected demand, which is continuing to increase plasma donations by 8 to 10% per year, we need to increase US plasma donations. And the best way to do this are the efforts taken by the IDF, the PPTA, and our own Facebook pages, social media, conversations with our friends, etc. We need to work towards allowing compensation of donors in other countries. So, for example, if we could have compensated donors in Canada or Brazil or more of Europe, we could get more plasma just like we have in the US. And we can optimize and innovate fractionation processes to increase the yield. So we have new entries into the market like the Evolve company, which is now building a plant in Texas. And again, the yield, the average yield of IgG now is about 3.5 grams from 1 liter of plasma, which is about 40 to 50% yield. And obviously everybody wants to increase this. Ways to mitigate or moderate the use is to increase the specificity of diagnoses, to not give huge doses of IgG for neuropathies that aren't going to respond, and to optimize the therapy for individual patients, as I mentioned. To the extent that other therapies such as FcRn antagonists, anti-B cells and anti-plasma cell therapies, and other biotech advances can substitute for IgG, then we'll have more IgG for those things in which there's nothing to substitute. The other side of the coin is, of course, everybody's working to improve the awareness and diagnosis of PID, and the increased use of anti-B cell and anti-plasma cell therapies for hematologic cancers are increasing the number and severity of patients with secondary immune deficiencies, which is increasing the demand for IgG. So we have some ways we can moderate the demand, but many ways that are increasing the demand. And the best market projections are that it will continue to stay about the same. So thanks very much. I think Tammy is going to present, and then we'll both take questions in a sort of panel situation. And if anybody wants to reach me offline, my email address is on the first slide. Thanks.
Thank you, Dr. Berger. As he stated, we will answer some questions after my presentation is complete. And I just have a few things that I want to share with you about what IDF is doing to ensure plasma supply as we move forward. So. It's obviously a critical relationship between plasma supply and our community. And what I'm going to talk about today is a little bit of the case for support that IDF is putting forward to just raise awareness about the critical issue of the need for plasma and getting members of the general community to understand what it is like to donate plasma and to celebrate plasma donors. And as part of that, we'll talk just a little bit about Plasma Hero. I'm going to talk a bit about what our public policy and advocacy team is doing in an effort to improve access to plasma, and then just close up with some recent wins, but also a little bit of the knowledge that we have a long way to go. So one of the things that we've been doing at IDF over the past several months is to really try and highlight the fact with the general public outside of our own community that there are more people that rely on plasma than many people think. And so this image that you're seeing is one that we've been using on social media. And when we meet with potential partners in the community, that there are enough plasma users in the US for chronic rare conditions that could fill a very large soccer or football stadium. And that does not count all the people that use it for burns, shock, emergency situations that they would use in a one-off time. The other things that we're doing is doing more on social media to talk about plasma education. And just this week, as we end International Plasma Awareness Week, there is a letter on our website from our CEO and President, Jory Berry, and the chair of our board of trustees, Tracy Shaw, about kind of where we are with plasma and IG access in this country, and a reassurance that we're going to always keep a close eye on it, and some good news that's come down the pike recently. Additionally, some of you may be aware, but I always like to mention it in case you're not. In 2020, IDF launched an initiative called Plasma Hero, which is a separately branded initiative and website that really, again, is the audience is the general public, and it is an opportunity to celebrate plasma donors for the heroes that they are, and to recruit new plasma donors to come out and decide to donate plasma and do it more than once. And some of these are the materials that we have and we're sharing. Kind of phase one of the project was when we first launched it, and we have a website and social media. This next year, we're going to start going out and doing some grassroots community outreach and speaking to public groups and college sororities and fraternities and workplaces and people that are looking for group volunteer opportunities to donate plasma. We are also going to be partnering with plasma centers directly so that we can give first-time donors a bit of a packet that says you donated plasma, you saved somebody's life, and let them kind of report that information to us so we can send them reminders, encourage them to set up another appointment, to bring a friend when they come the next time, and to hear stories from patients like all of you and how what they're doing is actually saving lives. Our public policy and advocacy team works really hard on a variety of different policy issues that affect our community. But one of the ones that's particularly relevant for us to talk about today is that they ensure access to care. So apologies, just going to read this because it's written better from our team than it would be for me to just verbalize it. IDF prioritizes advocacy efforts that ensure patients have ready and affordable access to health care in the site of care that is most appropriate with the treatment recommended by their medical providers. And in doing that, they obviously do a lot in the space of plasma access and awareness. And we do that work in many states. Different states have different regulations on what it takes to build a plasma center. And some of those regulations in some states are quite burdensome. California, in fact, just recently lifted some of those burdens in a way that's going to make it possible for new plasma centers to open in California. And so we're really excited about that. But that work is happening across the country. And then obviously, as there are ID access issues that happen for Medicare, private insurance, Medicaid, we're working hard to make sure that we can help with those access issues as well. And here's some recent good news that has been happening. And there has been a lot this summer. It's been a really active, busy summer in the IG plasma landscape. Some of you may or may not be aware that about a year ago, the federal government began denying B1 and B2 visa usage for Mexican citizens who had for years crossed the border into Texas to donate plasma at Texas donation centers. And a judge just in the last 2 weeks has put an injunction on that. So Mexican donors can again begin to donate plasma, which makes a huge difference to everyone, obviously. So that currently is temporary. Our team is certainly keeping an eye on that and seeing if that we can make sure that that injunction becomes permanent. So we will continue to update the community as that situation evolves. The U.S. Department of Health and Human Services launched a Giving Equals Living campaign, which is a national website that encourages both blood and plasma donation. But we have a member of our own community here at IDF who is one of the featured patients who tells her story via video and on the website. So I encourage you to look at it. The fact that there is some federal power behind that, I think, could really make a difference in plasma donations. As I mentioned, California Assembly Bill 392 lifted some of those regulations for building a plasma donation center. And so we really will be able to, I think, see a difference in what is obviously our most populated state. And there's a lot of potential access to plasma donors there. And then there are new technologies coming out every day that improve the plasma collection process that makes it speedier, less painful, more efficient, and more effective. That I think will, if the process is easier and better for plasma donors, they'll come back and donate more often, which we think is always, always good news. And then obviously we still have a long way to go. There are always concerns about plasma access. So I want to ensure you that our organization will always keep an eye on any potential shortages or delays in the supply chain. While parts of 2 Canadian provinces recently began allowing plasma centers to compensate donors for their time, most of the world still relies on American donors for sustainability. And that is something that we will always keep an eye on and insert any information that we can as possible. We can't always control what's happening in the rest of the world, but we always want to be a voice for plasma patients. And I want to just kind of end this with a reminder to all of you, if you ever have plasma access issues, please report them to Ask IDF. We always want to keep an eye on what's happening and when and how, and that is our best way to kind of make sure that we know what's going on so we can help you. And that is all I have to say about the IDF piece. I want to open it up for any questions that we have. And it looks like we have one question, Dr. Berger, that has already come in. And I want to encourage everybody to keep putting questions in the chat as you have them. But this first one is, could mRNA technology be used for IgG to teach the body to be able to make their own antibodies? Oops, sorry, without the need to use IgG every few weeks?
Well, the short answer is not really. Patients who require IgG on a regular basis don't make antibodies very well. And so mRNA, which is used now not to directly code for antibodies, but to code for antigens to act as a vaccine to induce an antibody response, we know that giving the antigen in that way does not work very well in people with serious impairments of antibody production, like some people with PID, obviously XLA, and we know that many patients with organ transplants fail to respond to those vaccines. So it's not a viable method of protein replacement.
Great, thank you, Dr. Berger. That's a great
Sure.
answer. It looks like the next question is for IDF, so I will try and address that. The question is, how can I get my local plasma donation center involved with the Packet Initiative? I volunteer at this center for IDF and want to involve them in this. Can I get a copy of the slides from this presentation? Absolutely. I'll answer the end of that question first. If there are not now, there will be slides to the presentation in the session attachments that you'll be able to see. And if not, I can always send them out to you. And on the getting involved, stay tuned. We'll have details for volunteers very soon, but you can always reach out to Ask IDF and they will know how to find me. And we can talk one-on-one on how you can help us get into those plasma centers that you know the managers of better than we do. Are there any other questions? I'd love to see if any other questions are coming in. We just have those two to get started, but we're happy to answer more. Oh, and my own colleague, Rachel Delaney, says, follow Plasma Hero on social media. Absolutely. And IDF, we do have separate social media for Plasma Hero. So it is Be a Plasma Hero on Instagram and Facebook. I'll pitch that a little bit. And we're everywhere as IDF on social media. So make sure you follow us there for sure. Here's another question. Looks like it is for you, Dr. Berger. Can you talk about the progress on fractionalization? Is anyone working on the strep P antibodies?
I'm sorry, I missed the last part of that.
Is anyone working on the strep P antibodies?
Well, I don't know exactly what they mean by the strep P antibody. But there certainly are small companies and also the big manufacturers trying to increase the yield of IgG per liter of plasma. And as I said, there's a company, which I forget now exactly what the product, I don't know the trade name. I don't know if the trade name of the product has been selected yet, but there have been successful clinical trials of IgG made from plasma, but by a completely different process, which takes less time and results in a higher yield. And I expect that we will be seeing higher yields coming from the major manufacturers also.
Great. Thank you, Dr. Berger. And I have a couple more questions that are specific to you and your expertise for sure. Do you think that FcRn antagonist will make patients more prone to more infections since it is decreasing IgG? We have seen that with rituximab, it can cause secondary immune deficiencies, thus needing IVIG.
Well, I think it will depend on the individual patient and the amount of autoantibody they make compared to the amount of protective antibody and the affinities of protective antibodies they make. But I certainly think it is an important point that the FcRn antagonists decrease normal antibodies by the same percentage as the autoantibodies, as the pathological antibodies. So although there have not been serious infections in the clinical trials to date, it's certainly something that will have to be kept under close observation. And it may limit the use of that form of therapy in some patients. And as you point out, this certainly limits the use of rituximab and the the anti-B cell therapies, because they certainly do increase the risk of infection. And in fact, they also increase the diagnosis of CVID in patients who were just presented with what they thought was autoimmune hemolytic anemia or autoimmune ITP. They give them Rituxan to treat that, and then they find out that the patient's own IgG never recovers because they actually had CVID all along.
Got it. We have a few people asking about what is probably no surprise to you, a hot topic. Do you think that plasma will ever catch up with the SARS-CoV-2 variants? When will we know if dates are not put on the plasma bottles? And will we, and it's a separate question, but same topic, will we ever be able to get enough antibodies to prevent COVID?
So I think there's two parts to the answer. First of all, we are already seeing increased amounts of antibodies in the donor plasma and in the IgG that is produced from donor plasma. And this results from widespread immunization and from people who have recovered getting better. So the IVIG or subcutaneous IgG that patients are getting now probably does offer some protection against COVID. Now, the other side of the question is what happened in the clinical trials with convalescent plasma. That's a complicated question because in most cases, the patients already had to have the virus and have symptoms before they were given the convalescent plasma. So preventing something with antibodies is much easier and better than treating it once you have it. And that's why it's difficult to interpret.
Got it. On the topic of convalescent plasma hyperimmunes, one viewer is asking, with the recent outbreak in New York, do you think there will be a role for hyperimmune for polio?
Well, standard IVIG by law has to be able to neutralize polio. That's one of the criteria for licensing and approving IgG. And I can remember as a kid, I frequently have talked about this. When I was a kid, before there was polio vaccine, if somebody on your street or in your camp or school bus got polio, everybody else got a whopping intramuscular shot of IgG, and it was no fun. So IgG is quite clearly, the polyclonal IgG is quite clearly effective at preventing polio and preventing measles. But again, you have to get the IgG before you're exposed.
Got it. That is all the questions from our audience. I'm going to wait just one more minute in case a couple more questions come in. And I have to say, Dr. Berger, I'm pretty impressed. Like, we're within a minute of our time. That's just amazing. I don't think we have any more questions. I want to thank you so much for joining us, Dr. Berger, and thank all of you who joined us today to watch this session on IgG and plasma. Thank you.